Now let's go to the glycosylation on the human receptors. As I mentioned earlier, the viral particles are coated with glycans, and the host receptors are also decorated or coated with glycans. So the receptor of SARS-CoV-2 viruses are human angiotensin converting enzyme. It is expressed on the surface of many cell types, particularly on lungs and intestinal tracts. Using spike protein on its surface, SARS-CoV-2 binds to ACE2 receptor, and that helps in the viral attachment and entry to human cells. Human angiotensin converting enzyme itself is glycosylated, and studying the glycosylation on them is critical for the viral infection. So the study of glycosylation or SARS-CoV-2 is important. Here in this figure you can see that the angiotensin converting enzymes contains different glycosylation sites, both N and O glycosylation sites, and those are dimer, a dimerized protein with 2 units, and SARS-CoV-2 RBD domain or receptor binding domain attached at this particular location where N90 and N322 are there. So studying this glycosylation will give more insights into how the viruses utilizes these glycans for the host attachment and entry. So this figure shows the experimental approaches that we used for the determination of glycosylation of human angiotensin converting enzymes. We employed through complementary techniques such as glycomics, that is the left panel, which includes the detailed analysis of glycans and its branching, and the right one is glycoproteomics, which determines the detailed information on glycopeptides, that means both peptides and the glycans. So you can see that the proteins will be denatured, as I mentioned earlier, then digested to several small peptides, and these peptides will be utilized further to release the glycans from them, and these glycans are later derivatized, as I will show later in another slide. Derivatization mostly we performed is permethylation, that means converting all hydroxyls on the glycans into methyl group. Then that will increase the mass spec sensitivity. Then they are analyzed by mass spec, either multi-MS or ESI-MS. So in the case of glycoproteomics, as I showed earlier, the protein or peptides are processed either directly or after processing through specific methods for the determination of their sites. Then these glycopeptides are analyzed by high resolution mass spectrometry, and the glycan structures on the peptides and their sites are determined. These are the glycans released from human angiotensin converting enzyme. They are characterized in detail by permethylation, as I mentioned earlier, and subsequent mass spectrometry analysis, such as matrix assisted laser desorption ionization or multi-MS. So the glycans will be released from the glycoproteins and then derivatized by methylation. That improves the sensitivity of glycan, and then we could see peaks like this. This is MS1, not MS2. So based on the MS1 mass, we annotate the glycan structures. So this figure shows the overall glycosylation profile that we obtained for angiotensin converting enzyme by glycoproteomics method. Here, in comparison to the previous figure, we have lot of site information. Earlier figure includes the total glycans, and this one will give a site specific glycan information. As I discussed earlier, the angiotensin converting enzyme is a dimer attached together, and there are a lot of N-glycosylations on that, and the spike protein binds to one particular region of the angiotensin converting enzyme. So the glycosylation at site N90 and 322 are most critical for the spike protein binding. So we deduced, or we studied this glycosylation at each of these sites as shown in these figures, and there are subsequent molecular biological and molecular modeling study which indeed proved that the N90 and 322 glycans are very critical for the viral attachments to human ACE2. Thank you, and please feel free to ask any questions. So these are the funding agencies that helped us in these studies.